Hi, I'm David, and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Table 1.1 shows the concentration of sodium and potassium ions inside the red blood cells and the blood plasma. A1 wants you to identify and describe the process by which potassium ions enter red blood cells from the blood plasma. The concentration inside the cell is much higher, so potassium ions cannot move in by any passive movement down the concentration gradient. They must enter by active transport so that they can move against the concentration gradient. Energy or ATP is required for the movement to occur, as this is not a random movement. A carrier protein must be used. When the ions binds to the binding site of the protein transporter, it undergoes conformational change. The binding site is specific for potassium ions. 2. State one similarity and one difference between the processes used by sodium ions and oxygen molecules to enter red blood cells. The concentration of sodium ions shown in the table indicates that they enter the red blood cell down the concentration gradient. Oxygen moves into the red blood cells down the concentration gradient too. This occurs near the lungs, where there is a high partial pressure of oxygen in the surrounding. No energy is needed for molecules to move down their concentration gradient. This is a random and passive movement. However, oxygen can move by simple diffusion, but sodium ions must enter by facilitated diffusion. This is because oxygen is small and uncharged. Ions are charged and hydrophilic. They cannot pass through the hydrophobic core of the phospholipid bilayer. A channel protein with a hydrophilic tunnel is involved in their movement. 3. Explain why the chloride shift is important in the transport of carbon dioxide from respiring tissues. When carbon dioxide produced by the respiring tissue enters red blood cells, it reacts with water to form carbonic acid. This reaction is catalyzed by carbonic anhydrase. It will then dissociate and form hydrogen carbonate ions and hydrogen ion. Hydrogen carbonate ions move out of red blood cells down their concentration gradient. They will be transported in the blood plasma. This movement causes a change of charge across the cell surface membrane. So, chloride ions, which are also negatively charged, move in to maintain the electrical neutrality inside the cytoplasm. Without the chloride shift, a negative charge builds up inside the cytoplasm. This can affect the electrochemical reactions inside the cell. There is a mark for AVP. For example, you can state that the exchange is important in maintaining the equilibrium. So, more carbon dioxide can enter the cell and react with water. B wants you to identify the process described in the question. When cell surface membranes invaginate and form vesicles to take up a substance, endocytosis occurs. You can also specifically identify it as pinocytosis, as this process does not involve the binding of a molecule to the receptor on the cell surface membrane. When a receptor is involved, it is phagocytosis. In question 2, we have an electron micrograph of an epithelial cell. A. Outline the role of structure X and structure Y in the production of extracellular proteins. Notice that the role you outline must be related to the production of proteins, so not all functions of the structures are accepted. X is a darker circular structure inside the nucleus, the nucleolus. It is involved in the synthesis of ribosomal subunits or ribosomes. Ribosome is the site of translation. Y points to the thread-like structure inside the nucleus, the chromatin. Chromatin contains genes coding for the polypeptides. Transcription occurs for the production of mRNA, which will be used later in the translation to synthesize polypeptides. B1. State the level of protein structure shown in figure 2.2. The diagram shows the sequence of amino acid in a polypeptide. This is the primary structure. 2 wants you to state one similarity between the structure of a saturated fatty acid molecule and an amino acid molecule. This is the general structure of amino acids and a saturated fatty acid. Both molecules have a carboxy group, the COOH, at one of their ends. Besides, they both have carbon, hydrogen, and oxygen. 3 suggests how the addition of this fatty acid chain allows a ghrelin molecule to act as a cell signaling molecule. A cell signaling molecule must have a specific shape complementary to the receptor. The fatty acid forms part of the overall 3D shape. Together with the polypeptide, they give rise to the tertiary structure of the molecule. Since fatty acid is hydrophobic, you can also say that it forms part of the hydrophobic region of the molecule. Having this specific 3D shape, ghrelin can bind to the receptor. It also allows ghrelin to have a complementary shape to the receptor, and this is the specificity of the cell signaling molecules. Due to its hydrophobic nature, it possibly interacts with the hydrophobic region of the receptor molecule for binding. It may position ghrelin better for the binding to take place. There is a mark for AVP. For example, you can talk about the consequences of the binding or provide some logical suggestions on how this fatty acid can help with the binding or the transportation of molecule in blood plasma. C wants you to calculate the percentage base pairs found in the gene that codes for ghrelin. There are 28 amino acids in the diagram. But don't forget that every gene must have a start codon that codes for methionine. Since the first amino acid in the picture is not methionine, the start codon is not shown here. You also have to add the stop codon, which does not code for any amino acids. Altogether, we have 30 codons. Each codon is 3 base pairs in the DNA. This means there are 90 base pairs in the gene. This is 1.8% of the total of 5,000. 2. Explain how the primary transcript is modified before translation. In our DNA, there are introns, the non-coding regions, and exons, the coding regions. All of them are transcribed into the primary transcript. Introns will be removed during the post-transcriptional modification, as they do not code for amino acids. Exons are joined together to form the mature mRNA. This process is called the gene splicing or RNA splicing. There is a mark for AVP. If you know about the other modifications that are not in the syllabus, you can get a mark as well. 3a talks about 2 types of cells in a tissue adapted for transporting assimilates. The tissue is phloem. The 2 cells that make up this tissue are the sieve tube element and the companion cell. Cell type A is the sieve tube element. We can tell from the fact that it has no nucleus. This feature maximizes the space available for the transportation of assimilates in their lumens. Plasmodesmata are found between the sieve tube element and the companion cell. They are microscopic channels that connect the plant cells, allowing for the exchange of molecules and communication between adjacent cells. In this case, plasmodesmata connect the cytoplasm of the 2 cells in the phloem. Assimilates can move from the companion cell into the sieve tube element or vice versa via the Syn+ pathway. This allows a rapid diffusion of sucrose from one cell to adjacent cell since they do not have to pass through the cell surface membrane. The efficiency of loading and unloading sucrose from sieve tubes has increased as there is a higher diffusion rate. In B, we have a diagram of cellulose. You are asked to complete the table to compare cellulose with cellulose. The monomer in cellulose is beta-glucose. The glycosidic bond joining the cellulose monomers occurs between carbon number 1 and 3. So it is called 1,3 glycosidic bond. A cellulose molecule is a straight-chain polymer. It is linear and unbranched. By looking at the positions of oxygen in the ring or carbon number 6, we can tell that all the monomers orientated the same way up. C. Describe the roles of hydrogen bonding in the movement of water through xylem vessels. This question specifically asks about the movement through the xylem vessels. So, your answer must only relate to this. Hydrogen bonding leads to 2 important forces, adhesion and cohesion. Adhesion resulted from the hydrogen bonding between water molecules and the cellulose cell wall. It can also occur with the hydrophilic parts of lignin in the cell wall of xylem vessels. Cohesion is the attraction between water molecules. It occurs because of the dipole property of water. Each molecule has a partial negative charge near the oxygen atom and a partial positive charge near the hydrogen atoms. The 2 forces help the formation of a continuous column of water. This causes a steady flow of water from the roots to the leaves. This steady upward movement of water is called the transpiration pool. A detail of the cause of transpiration pool will give you a mark. For example, state that the evaporation of water occurs on the spongy mesophyll surfaces. There is a mark for AVP. You can talk about how a hydrogen bond forms between two water molecules and leads to cohesion. D, with reference to hydrogen bonding, suggests why cooling of the leaf occurs as a result of evaporation of water during transpiration. For water to change its state from liquid to gas, sufficient heat energy must be absorbed to break the hydrogen bonding between them. Water has a high latent heat of vaporization. It requires a large quantity of thermal energy to change from liquid to gas. So, when water evaporates during transpiration, it absorbs a lot of heat energy from the plant. This causes the plant to lose heat and cool down. Figure 4.1 shows some statistics about new HIV infections around the world. The full name of the pathogen is human immunodeficiency virus. A2 wants you to state what can be concluded about the change in number of new HIV infections across the world between 2010 and 2018. Only 2 regions show an increase. We can conclude that there is a decrease in the percentage of infections in most regions. The only exceptions to this decreasing trend are regions B and D. State the greatest and smallest decreases can give you 2 marks. You can quote the data to support any point you have mentioned. For example, you can quote the percentage decrease for regions B and D to support the second point. Remember to place the data right after the point it supports. Do not write it after you have made all the statements. There is 1 mark for AVP. For instance, you can state that even though region E shows the greatest percentage decrease, but it has the highest number of new cases. B. Explain why people with HIV/AIDS are more likely to develop TB. TB is an opportunistic disease for HIV patients. Opportunistic diseases or infections are infections that occur more frequently or are more severe in people with weakened immune systems, including HIV. Then, provide the details of how HIV weakens a person's immune system. The virus targets T helper cells. People living with HIV may have fewer T helper cells, hence a less effective primary immune response. Helper cells specialize in secreting a cell signaling molecule called cytokine. When their number decreases, less cytokines are secreted. Cytokines stimulate the immune system by signaling other cells. With lesser cytokine, fewer phagocytes are activated to perform a more vigorous phagocytosis. Besides, fewer B lymphocytes are stimulated. A smaller number of B cells will divide by mitosis and form plasma cells. When the number of plasma cells is small, fewer antibodies are secreted, so fewer pathogens are destroyed. People with a healthy immune system may not develop symptoms even if they are infected with Mycobacterium tuberculosis. This is called latent TB. As HIV weakens a person's immune system, latent TB may become activated. There is a mark for AVP. You can talk about how a weakened immune system affects the effectiveness of the TB vaccine or name the pathogen that causes TB. C shows a condition known as cardiac tamponade. It causes the pericardium to fill with blood. The question wants you to suggest why a person with cardiac tamponade would experience symptoms of low blood pressure and an increased breathing rate. The pericardium is supposed to be flexible and stretchy. It can easily expand and contract following the heart's contraction and relaxation. When it is filled with blood, it causes compression to the ventricles. So, the volume of the ventricles reduces. They can no longer fully increase in size. With less blood filling the ventricles, less blood will leave the ventricles in each cardiac cycle during ventricular systole. Ventricular systole will become less powerful. The volume of blood pumped to the pulmonary and systemic circulation per contraction will decrease. In other words, the cardiac output decreases. Lesser blood transported to the lungs causes less oxygen to diffuse into the blood and be delivered to the tissues. A faster breathing rate occurs to increase the quantity of oxygen absorbed in the lungs. This is to compensate for the lower concentration of oxygen in the blood. A higher breathing rate also increases the quantity of carbon dioxide excreted. There is a mark for AVP. For example, you can link the increased breathing rate to the cardiac muscle not receiving enough oxygen. You can also state that the respiratory center in the brain is responsible for the faster breathing rate. Figure 5.1 is a photomicrograph of a bronchus. The tissue labeled T is the cartilage. Angular cells called chondrocytes can be found in groups of 2 or more in the cavities called cartilage lacunae. 2. Describe the function of smooth muscle in the bronchus. Smooth muscles can contract and relax. They do this to change the diameter of the lumen. When they contract, the lumen size decreases, and when they relax, the opposite happens. This is done to regulate the airflow into and away from the gas exchange surfaces. There is a mark for AVP. For example, you can state the body condition when smooth muscles need to contract or relax. Note that you won't be credited if you use the term stretch and recoil instead of contract and relax. Those words describe the function of elastic tissue, not the smooth muscle. In B, a diagram shows type 2 epithelial cells secreting surfactant by exocytosis. Surfactants lower the surface tension and prevent the alveoli in the lungs from collapsing when we exhale. One wants you to draw a diagram to show the arrangement of phospholipid molecules in the membrane. Your drawing should contain a few phospholipid molecules, each with a head and 2 tails. They arrange into a bilayer with their tails facing each other. 2. Suggest why secretion of surfactant is affected by a reduction in cholesterol in the cell surface membranes of type 2 epithelial cells. The question mentioned that surfactant is released by exocytosis. This involves the fusion of the secretory vesicle called lamellar body with the cell surface membrane. Cholesterol regulates the fluidity of the membrane. When cholesterol decreases, the fluidity of the membrane decreases. With reduced fluidity, fusion between the lamellar body and the cell surface membrane is more difficult to occur. There is a mark for AVP. You can talk about how reducing cholesterol can decrease membrane fluidity. 3. Suggest why macrophages engulf surfactant. Macrophages engulf unwanted or foreign substances in our bodies. In this case, they help to prevent too much surfactant from building up in the lungs. Following endocytosis, lysosomes in the macrophage release hydrolytic enzymes to digest the surfactant. Products of the hydrolysis can be reused or recycled. Macrophages can use molecules such as protein, phospholipid, and cholesterol in surfactants to synthesize their structures or other substances. Pathogens may be trapped in the surfactant. Macrophages engulf the trapped pathogens and destroy them as a defense mechanism. There is a mark for AVP. For example, you can state the negative impact of having too much surfactant in the alveoli on gas exchange. DNA replication occurs during the S phase. It is a stage in the interphase. Figures 6.1 and 6.2 show a normal nucleotide and a modified nucleotide that can prevent the elongation of polynucleotide. You are asked to suggest how the structure of the modified nucleotide prevents DNA polymerase from joining it to another nucleotide. Two nucleotides are joined together by a phosphodiester bond, which is formed through a condensation reaction. The reaction occurs between the OH on the phosphate group of one nucleotide and the OH on carbon 3 of the deoxyribose in the next nucleotide. The diagram shows that the modified nucleotides does not have an OH group at carbon number 3. Without this OH group, a condensation reaction cannot occur. So, phosphodiester bond cannot form between deoxyribose and phosphate of the adjacent nucleotide. Another reason is that it has a different shape from a normal nucleotide as it has a H instead of OH. So, its shape is not complementary to the active site of DNA polymerase, the enzyme that catalyzes the formation of phosphodiester bonds between DNA nucleotides. No enzyme substrate complexes can form for elongation to take place. Table 6.1 shows the effect of two RNA aptamers on the activity of RNA polymerase. B1 wants you to compare the effect of the aptamers on the affinity of RNA polymerase for its substrate. To comment on the affinity of an enzyme, you need to look at the Km value. The Km value, or Michaelis-Menten constant, is the concentration of a substrate at which an enzyme's reaction reaches half of its maximum velocity. If the Km value is higher, a greater concentration of substrate is required to reach half of Vmax. This means the enzyme does not bind so well with the substrate. It is said to have a lower affinity. Both aptamers increase the Km of the enzyme. This means that both reduce the affinity of RNA polymerase for its substrate. However, F52 increases the Km value more than F47. This indicates that it reduces the affinity for the substrate more than F47. There is a mark for AVP. You can manipulate data from the table to support any described trend. For example, you can compare the increase of Km values by the aptamers. You can also state the relationship between Km values and affinity. 2. Suggest explanations for the effect of the presence of an aptamer on the rate of transcription catalyzed by the RNA polymerase. The question wants you to discuss the rate of transcription. So, you should look at Vmax, the maximum velocity or rate of reaction. Vmax of the enzyme is lower in the presence of aptamers. This indicates that the aptamers may be inhibitors. There are a few possible ways they might work. They can bind to the enzyme and change the tertiary structure of the enzyme. This causes the shape of the RNA polymerase active site to change. The substrate, RNA nucleotides, can bind less easily to the active site. This leads to fewer enzyme-substrate complexes formed. The binding may also block the active site. This prevents the substrate from binding to the enzyme's active site. Another possibility is that the binding prevents RNA polymerase from binding to the template strain. Transcription cannot be initiated if the RNA polymerase does not bind to the DNA template. The binding can prevent RNA polymerase from functioning normally. It can no longer catalyze the forming of phosphodiester bonds between the RNA nucleotides. There is a mark for AVP. For example, aptamers bind to allosteric sites on RNA polymerase. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.